Hi, welcome to this talk entitled Regional Genetic Diversity in Spartina and Juncus, Implications for Future Salt Marsh Restoration Success. This work was conducted by myself and my colleagues Mark Welch, Brian Baldwin, and Jesse Morrison at the Mississippi State University. A brief overview of what we will cover today. I will talk about salt marsh restoration and the need for plants, genetic diversity in restoration, the various components of the research spread across 3 years, and finally wrap up with the implications of this research. Restoration in the northern Gulf of Mexico, in particular in coastal Mississippi and Alabama, has undergone a dramatic increase since the 2010 Deepwater Horizon oil spill, which resulted in a $20 billion settlement between BP and the federal government in 2016. This money is being dispersed through various pathways, including Restore, National Resource Damage Assessment, and National Federation of Wildlife and Fish. Beneficial use of dredge spoil for salt marsh creation is a primary activity in the state of Mississippi. Gulf-wide, there are over 13,000 acres of marsh restoration that are worth at least $175 million already in progress, and this number is now a couple of years old. Planting typically requires between 5,000 to 45,000 plants per acre, depending on the spacing, so a very large number of plants need to be readily available. What are the goals of salt marsh restoration? Well, it depends a bit on where you are in the process of doing this. For instance, if you are providing sediment through dredge spoil and beneficial use, then you may have the end goal of having that sediment become emergent and at the right elevation for the colonization by salt marsh plants. Alternatively, if you are into vegetation primary production, your goal may be the planting at the site once the elevation is appropriate. Finally, over time, the vegetation does coalesce and more species come in through natural recruitment until you have a broadly vegetated habitat which may or may not be analogous to a natural salt marsh. In addition, just because it looks like a marsh doesn't necessarily mean that it functions like a marsh. These various time steps are part of the successional process after the marsh has been created. And one of the things that I'd like to address today is the need to consider genetic diversity as part of the process of developing and executing that restoration. Now, genetic diversity is important in a natural population to increase resilience to environmental change, disease, and other factors that may differentially affect different genotypes. In agriculture and plant production, generally, we tend to focus on developing the best varietal or cultivar. That is, we achieve a minimum genetic diversity so that everything can look and grow the same. I would argue in restoration that we are intending to maximize genetic diversity so that the population will remain resilient to future change. And because of this, the restoration needs are in opposite to the long-standing plant propagation and selection practices that we have in agriculture. Let's look at salt marsh plant diversity in particular. The two species we looked at were smooth cordgrass and black needlerush. There are many prior studies from Louisiana as well as elsewhere along the Gulf of Mexico. Many of these are relatively small-scale genetic assessments. Many of the published works focus on smooth cordgrass, Spartina alterniflora, with much less information available on black needlerush, Juncus rhamerianus. The United States Department of Agriculture Golden Meadows Plant Materials Center has an approved cultivar for Louisiana for Spartina alterniflora, but this species does have low genetic diversity and often poor seed set. There is currently no approved cultivar for Juncus rhamerianus. However, black needlerush is the dominant species throughout much of the northern Gulf of Mexico, including Mississippi and Alabama. And few salt marsh studies in either species have looked at genetic diversity over a larger regional scale on the order of hundreds of miles. So we set out to investigate the genetic diversity of these two species within Mississippi as well as in neighboring states and sampled over about 150 miles of shoreline from Lake Pontchartrain, Louisiana to Pensacola Bay, Florida at these 10 locations. Here's a map showing Fork Pike on the western side of our sampling effort near Slidell, Louisiana, and Point Garcon all the way to the east near the city of Pensacola, with the additional 8 sites in between. During the collection phase, we targeted 10 locations during the summer of 2016 and generally collected from 1 or 2 sites per day. And you can see the site in the background looks very typical of where we collected, with Spartina alterniflora being the brighter green grass growing closer to the water's edge, and Juncus romarianus being the darker olive gray rush that's growing a little bit upslope and to the left. The minimum distance between sites was 6.5 miles. The maximum distance was 45 miles. At each station, we collected 25 individuals of each species for a total of 50 individuals in the 2 species. Approximately 6-inch diameter plugs were extracted, individually bagged and transported back to the lab, where we potted them in 1-to-1 sand-soil mix in trade-gallon pots, sub-irrigated them in the greenhouse for a number of months to recover from collection stress before transferring them to Mississippi State for the genetics analysis. At MSU, the salt Martians got to work doing the genetics of these 2 species. DNA extraction was done using Promega Maxwell. PCR amplification was used based on published primers for both species, and it's important to note that Tumas et al. Were the first published primers for Juncus romarianus. Spartina had published primers that were older than that. Finally, fragment analysis was done at Arizona State University's DNA lab. And data analysis was then done on the information using Peak Scanner Gene Marker and GenLX software. I will move on to some results now. The first figure here shows the sites along the bottom axis, with Fort Pike, Louisiana on the left. That's the westernmost site, and then heading eastwards to Pensacola Bay, Florida. The 6 stations in the gray box are all within the state of Mississippi, and we can see the allelic richness for Juncus in red tends to be higher within Mississippi than in the stations outside, and the allelic number of alleles for Spartina alterniflora in blue tends to be relatively high in Mississippi, but also has some high stations in Alabama. The effective population size now, same stations from left to right, again within the state of Mississippi. The Juncus tended to have a larger effective population size, whereas Spartina tended to have a larger effective population size as we moved eastwards out of Mississippi through Alabama and Florida. We then looked at linkage disequilibrium to get an understanding of some of that allelic diversity and recent evolutionary change. The numbers here, blue indicate smaller values, red indicate larger values. Larger values are considered to be a poorer population in terms of diversity. Here we can see the map showing Graham Bay and Pops Ferry tended to have the highest linkage disequilibrium for Spartina alterniflora, and LD was lower as you moved further east towards Pensacola Bay. The results for Juncus show that Weeks Bay and Heron Bay around Mobile Bay proper had the highest linkage disequilibrium, and it was low in most of the other sites. Here are a summary of the 3 results showing the 2 species on the left, with the populations broken up for those found within the state of Mississippi in the gray rows and the non-Mississippi populations in the white rows. We can see for allelic richness, both species had higher richness in the state of Mississippi than outside. For effective population size and linkage disequilibrium, the pattern for the 2 species was different. In Spartina, those 2 tended to be higher outside of the state and mostly towards the eastern sites. In Juncus, both of those results tended to be highest within the state. What are the implications of this research? Genetic diversity based on these 3 metrics was found to differ between Spartina and Juncus at the regional scale. Regional diversity was more variable in Spartina across the 10 sites, whereas Juncus had the higher diversity within the sites in Mississippi than those outside of Mississippi. Future restoration projects can be composed of diverse germplasm lines, i.e. collected from multiple different locations, that when planted together have a long-term benefit. This is something that we are still investigating with the plants that were collected. Implications for other restoration practitioners should be asking the nursery what methods were used in propagation of plants. Were these field harvested? Were they a small population that was continually propagated through clonal propagation? And what might be the implications of that on the diversity of the plants that are then planted into the habitat? In addition, many times nurseries are located far from the source of where the project is going to occur, and so we should expect different genotypes to be entering the restoration population because of this relatively long distance transport of the nursery raised individuals. Thank you for your time and attention to this talk. You can contact me at my email address, patrick.biber@usm.edu, for more information. I want to acknowledge the Mississippi Department of Marine Resources for providing the funding to do this research. And if you're interested, you can look at additional details on this project at my website, Marsh Genetics. Thank you very much, and I look forward to the question session.